On behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor, MaxCyte, I'd like to welcome you to Accelerating Product Development, from transient transfection to stable pools and stable clones. My name is Elizabeth Lamb, and I'll be your host and the moderator for today's event. I'd like to introduce our presenter for today's webinar. James Brady, PhD, is Director of Technical Applications at MaxCyte Incorporated, where he's worked since 2004. Prior to joining MaxCyte, Dr. Brady was a senior scientist at Genetic Therapy Incorporated, which is a Novartis subsidiary, and a group leader at Metamorphics Incorporated. Dr. Brady earned a Bachelor of Science in Biology from the College of William and Mary and a PhD in Genetics from Indiana University. He also received postdoctoral training in the National Eye Institute of the National Institutes of Health. Our second speaker is Jerome Cortet, joined Valneva, formerly known as Vivalis, in July 2009 as a project leader working on the development of the EBX platform for the production of therapeutic monoclonal antibodies. Since November 2012, he's been managing the Research and Development Protein Production Group, specifically in charge of the production, purification, and characterization of recombinant proteins on CHO-based expression platforms to support clients' projects or internal requirements. The Valneva CHO platform has a robust and reliable process for generating stable clones expressing recombinant proteins up to bioreactor levels. In addition, Valneva has developed a production process for large-scale transient expression based on the MaxCyte STX technology that enables a quick response to internal requirements, but also and especially to those from the VIVA screen, the fully human monoclonal antibody discovery platform. Our third speaker is Audrey Angel, who is the Research and Development Engineer at Valneva. She joined Valneva in 2008 after receiving her Master of Science in Biotechnology from ESBS and 3 years in academic research in Bern, Germany. Ms. Angel has worked on the development of an automated process for EB66 cell line selection prior to contributing to Valneva's CHO platform creation. After implementation of a stable cell line selection process, she's now involved in transient protein production optimization for customer and internal projects, supporting the VivaScreen antibody discovery technology platform. With that, James, I'll pass you the presenter ball. Welcome.
Great. Thank you, Elizabeth. I'd like to start by welcoming all of you to our webinar. Today, I'll be highlighting the advantages of MaxCyte's scalable electroporation-based transfection technology for antibody production in CHO cells. And I'm pleased to be joined by Jerome Corteta and Audrey Angel from Valneva, who will share their experiences with MaxCyte technology for protein expression in CHO cells. This is an outline of the topics that I'll cover in my portion of the webinar. I'll begin by highlighting key advantages of MaxCyte's technology for streamlining the development of antibody therapeutics and other protein products. To place MaxCyte's technology in context, I'll provide a brief overview of the challenges that are associated with antibody development, and I'll discuss the advantages of large-scale transient transfection in CHO cells for accelerating development timelines and for overcoming hurdles in the development process. Next, I'll give you an introduction to MaxCyte's technology, highlighting key attributes such as scalability, reproducibility, and compatibility with many different cell types. For the remainder of my portion of the webinar, I'll share data on antibody production in CHO cells following transfection with MaxCyte's flow electroporation technology. I'll present data illustrating the high titers that can be achieved via transient expression in CHO cells, seamless scalability of the MaxCyte process, advantages for streamlining upstream culture and downstream processing, applicability for producing non-antibody proteins, and finally, benefits of MaxCyte electroporation for generating stable pools and stable clones. This slide presents the key messages that we'd like you to take away from today's webinar. MaxCyte's scalable transfection technology transfects CHO cells with high efficiency and high viability, allowing users to screen a large pool of antibody candidates via transient expression in the same cell background that ultimately will be used for biomanufacturing. MaxCyte technology does not place significant constraints on cell density, which enables process streamlining and conservation of resources. As I mentioned earlier, all preclinical work can be done in the same CHO cell background that will be used for production of the final protein product. High transfection efficiency and seamless scalability enable production of multi-gram quantities of antibody product from a single CHO cell transfection using our STX instrument, and with our VLX instrument, we offer the potential to produce up to 50 grams of protein at one time by transient expression. While expressing proteins following a large-scale electroporation, some of the transfected cells can be set aside for selection of stable clones. Thus, MaxCyte technology offers the potential to generate milligram to gram quantities of protein very quickly for initial analysis, while enabling researchers to get a head start on generation of stable cell lines for advanced development work. The bottom line is that all of the above advantages add up to significant time and cost savings. And this is a point I'll raise multiple times throughout my presentation. By way of background, I want to spend a few minutes focusing on the steps that most companies go through to develop an antibody therapeutic. After identifying a therapeutic target, the first step is to generate a pool of antibody candidates that bind to the target using phage display or related techniques. The resulting pool of antibodies needs to be screened to identify more promising candidates based on binding affinity, biological activity, or other relevant metrics. And at this stage, when milligram quantities of protein are required for analytical activities, most researchers resort to expressing proteins via transient expression in HEK cells, primarily because HEK cells are easier to transfect using conventional transfection methods. After the pool of candidate antibodies has been narrowed down, larger quantities of protein are needed for more detailed studies on pharmacology, toxicity, formulation, etc. Here, it's really important to generate protein in the same CHO cell background that ultimately will be used for manufacturing. Therefore, at this stage in the development process, researchers generally resort to generating stable CHO cell lines to produce the quantities of protein required for advanced preclinical studies. Finally, a single candidate antibody then is selected for production in a stably expressing CHO cell line. So there are two major problems with the traditional approach to antibody development. First, by expressing proteins transiently in HEK cells, differences in post-translational modification and protein stability relative to proteins produced in CHO cells may confound interpretation of bioefficacy data. In addition, biomanufacturing challenges may not become evident until late in the development process. On the other hand, producing all candidate antibodies in CHO cells becomes a challenge due to the fact that most conventional transient transfection technologies work poorly in CHO cells, and generation of multiple stable CHO cell lines requires a significant commitment of time and resources. Ideally, the middle two steps in this traditional process should be eliminated and replaced by a methodology that allows large numbers of antibody candidates to be screened via large-scale transient expression in the same CHO cell background that's going to be used for manufacturing. The transient expression technology that I proposed in the previous slide must meet a number of criteria. First, it should be compatible with many different cell types that are used for biomanufacturing, not just CHO, but also HEK, Vero, CAP-T, BHK21, and even insect cells. Second, quality is critical. This means producing high levels of transfection efficiency while maintaining high cell viability. Another key requirement is scalability. To generate multi-gram or milligram to gram quantities of protein, the technology should be able to transfect anywhere from a few million cells up to billions of cells with the same levels of efficiency. The technology should be rapid and easy to use while providing consistent results. And finally, it should enable the expression of multiple types of proteins, including secreted proteins, membrane proteins, multi-subunit proteins, and viral vectors or VLPs. The technology that meets all these criteria will be extremely cost-effective because it will greatly reduce project timelines, it will lower running costs, and minimize required resources. This is where MaxCyte comes into the picture. In the next few slides, I want to give you an overview of MaxCyte's scalable electroporation-based cell loading technology. I'll focus on the seamless scalability of our process, highlight reproducibility and consistency of the transfection results, and describe the many different cell types that can be transfected at high efficiency while using the MaxCyte system. This slide highlights key attributes of MaxCyte's technology. First, with most cell types, we can achieve greater than 90% transfection efficiency while maintaining greater than 90% cell viability. The electroporation-based technology can transfect and co-transfect a wide variety of loading agents, including plasmid DNA, messenger RNA, siRNA, proteins, and cell lysates. Temporary openings are introduced into the cell membrane by exposing the cell to a brief series of electrical pulses, so any type of molecule can be introduced into the cell by this process. No adjustments to the electrical parameters are required to accommodate different loading agents. The technology is compatible with a wide variety of cell lines as well as primary cells and stem cells. One of the most unique features of the MaxCyte platform is its scalability. The STX instrument can transfect as few as 500,000 cells or up to 2 × 10 to the 10th CHO cells in a single transfection. Smaller numbers of cells are transfected in a cuvette-style chamber via static electroporation. At this scale, the process takes only a few seconds. Larger numbers of cells can be transfected by flow electroporation using the same instrument. The maximum time for a flow electroporation is less than 30 minutes. Both the static and flow electroporations take place in sterile, single-use processing assemblies, so there's no risk of contamination from one transfection to the next. The different-sized assemblies which are used with the STX instrument, including the cuvette-style OC100 and OC400 chambers, their intermediate-scale CL1.1, and the CL2 assembly, which is used for flow electroporation, are shown at the bottom of the slide. Again, all are single-use and disposable to maintain sterility. In addition to the STX instrument, which can transfect up to 2 × 10 to the 10th CHO cells at one time, MaxCyte also offers a larger scale instrument, the VLX, which can transfect up to 2 × 10 to the 11th CHO cells at one time. As you can see in this slide, both are benchtop instruments that fit easily into most research or production facilities. Those of you who are already familiar with MaxCyte's technology may notice the STX instrument has undergone a design change for 2014. The 2014 STX has all of the same features as earlier versions of our instrument, including the same software interface. Performance also remains unchanged. The only difference is a reduced benchtop footprint. Now I want to take a few minutes to walk you through the MaxCyte transfection process. To begin with, cells are harvested from conventional culture vessels. No specialized vessels, media, or culture protocols are required for transfection with the MaxCyte system. After a brief centrifugation step to remove the culture medium, cells are suspended at high density in MaxCyte's electroporation buffer. The buffer is a salt solution which contains no proteins or biological agents. We use the same buffer for every cell type. CHO cells are typically suspended in buffer at a density of 2 × 10 to the 8 cells per mil. Then at this stage, loading agents are added to the cells. For protein production in CHO cells, typical DNA concentrations range from 1 to 2 micrograms per million cells, which is consistent with DNA concentrations required for other transient transfection methods. The next step is to transfer cells and loading agents into the processing assembly. This step is performed in the hood to maintain sterility. The closed processing assembly containing cells can then be removed from the hood and attached to the STX or VLX instrument, depending on the scale of transfection. Remember, the STX can transfect between 500,000 and 20 billion CHO cells. The VLX can transfect up to 200 billion or 2 × 10 to the 11th CHO cells at one time. After attaching the processing assembly to the instrument, the user selects a pre-optimized protocol from the drop-down menu in the software interface. The user doesn't make any changes to the electrical parameters. The final step is click the Start button. Static transfections in the OC100, OC400, and CL1.1 processing assemblies take place in a few seconds. Flow electroporation in the CL2 and VLX disposable chambers takes place in less than 30 minutes. After the cells are electroporated, the processing assembly is taken back into the hood, transfected cells are removed, and then following a 30 to 40 minute recovery period, they're returned to culture, again using conventional culture vessels, media, and protocols. I should point out that a lot of our customers use the STX to generate cells for cell-based assays. As with protein production, the STX provides a rapid and flexible alternative to stable cell line generation for assays. After transfecting cells by flow electroporation with reporter molecules or drug targets such as GPCRs and ion channels, the cells can be aliquoted and cryopreserved. This allows users to thaw cells whenever they want to run an assay, and because they have multiple vials derived from a single large-scale electroporation, they get very consistent results in each assay run. As I just mentioned, users don't make any changes to the electrical parameters. They simply select a protocol optimized for their cell type from a drop-down menu. This is our current standard protocol list, which contains optimized protocols for over 60 cell types. If your cell type of interest is not on this list, we also provide 10 optimization protocols ranked from low to high energy that should allow you to easily find conditions that provide good transfection efficiency with your cells. And in this list, I've also highlighted cells in blue that are commonly used for bioproduction. In this slide, I'm showing you pictures of cells that we transfected with GFP plasmid DNA using the MaxCyte STX, just to give you a visual representation of the high levels of efficiency and viability that we can achieve via electroporation with plasmid DNA. Again, cells used for bioproduction are shown in blue. And note that the technology also provides highly efficient transfection with insect cells, such as SF9s. MaxCyte eliminates the need to generate baculovirus in order to introduce genes into insect cells. In addition to saving time and effort, removing the need for baculovirus transduction eliminates the potential for contamination of your expressed proteins and VLPs with baculovirus particles. For the remainder of my portion of the webinar, I'd like to focus on advantages of MaxCyte technology for protein production in CHO cells. I'll share data on the high antibody titers that could be achieved following transient transfection of CHO cells. I'll also highlight advantages for process scalability and process streamlining. I'll show some data with non-antibody proteins, and finally touch on ways that MaxCyte can reduce the time and effort required to generate stable pools and stable clones. By the way, I see a number of questions are appearing on our chat menu. We'll try to get to as many of those as we can at the end of the seminar. For the remainder of my portion of the webinar, I'd like to focus on advantages of MaxCyte technology for protein production in CHO cells. I'll share data on the high antibody titers that can be achieved following transient transfection, and I'll highlight the advantages for process scalability and process streamlining. So here at the top of this slide, you can see pictures of CHO S cells that were electroporated with GFP plasmid DNA using the MaxCyte STX. 48 hours after transfection, almost all of the cells were expressing high levels of GFP, and viability was close to 100%. These levels of efficiency and viability are typically much higher than can be achieved with other conventional transfection methods. High transfection efficiency and high viability translate to high productivity when CHO cells are transfected with the same concentration of antibody expression plasmid. At the bottom of the slide, you can see that titers of more than 1 gram per liter were achieved via transient expression following electroporation of CHO S cells with a bicistronic plasmid encoding human IgG1 heavy and light chain proteins. Here are some data on the seamless scalability of transient antibody expression in CHO cells following MaxCyte electroporation. In this experiment, CHO S cells were transfected with a human IgG1 expression plasmid at small scale via static electroporation in an OC400 processing assembly, and at large scale via flow electroporation in a CL2 processing assembly. Both sets of cells were seated in a shake flask at similar concentrations and cultured under identical conditions. You can see that the small-scale electroporation with 80 million cells and the large-scale electroporation with 10 to the 10th cells produced consistent antibody titers with comparable expression kinetics. Note that both sets of transfected cells yielded titers greater than 1.2 grams per liter. So in this case, by using flow electroporation, more than 3 grams of protein could be produced in a 3-liter shake flask. To highlight the reproducibility and consistency of MaxCyte's transfection process, we compiled data from 10 independent transfection experiments performed on different days via either small-scale or large-scale electroporation. In each experiment, CHO S cells were transfected with an identical concentration of a human IgG1 plasmid and seated in the shake flask at similar densities. Cultures were maintained until viability dropped below 50%. You can see that the volumetric titers and specific productivity numbers in the last 2 columns are very consistent among the different sets of transfected cells. Because we use the same electrical parameters, for each electroporation, transfection efficiency and viability are typically much more consistent relative to chemical-based transfection methods. Another important advantage of the MaxCyte process is that it does not place significant restrictions on culture density either before or after electroporation. So again, this contrasts with chemical-based transfection methods, which typically require you to keep the cells at about 1 million cells per mil at the time of transfection. So to illustrate this point, we harvested 2 sets of CHO-S cells for electroporation when their densities were either 4 million per mil or 8 million per mil, respectively. Both sets of cells were electroporated with a human IgG1 plasmid and then seeded back into separate shake flasks at densities around 4 to 5 million cells per mil. We cultured the cells for 13 days, and then we measured secreted antibody titers at 3 time points. So at the bottom of the slide, you can see the cell density data and the increasing titers in the 2 flasks. You can see you have consistent growth curves as well as protein production levels in both sets of transfected cells. And you can see that cells, even the cells that were 8 million cells per mil prior to transfection, this high cell density caused no appreciable loss of productivity. Now, if you extrapolate from these data, the cells are cultured at a density of 5 million cells per mil before electroporation, as well as after electroporation. 10 billion cells, 10 to the 10th cells, can be maintained in a 2-liter culture, and based on the final titer shown here, more than 1 gram of antibody can be generated in a single shake flask. Thus, with the MaxCyte process, there's no need to employ bulky wave bags or bioreactors to produce large amounts of protein. Therefore, MaxCyte minimizes reagent consumption, it simplifies cell culture processes, and it streamlines downstream purification. All of these benefits add up to significant time and cost savings. I should point out that to maintain cells at high density for extended periods of culture, we use a batch-fed process. The feed supplements that we use are all commercially available. In addition, we implement a temperature shift of 32 degrees 1 day after transfection. This also is a very common practice in the protein production community. Here are data from Lake Pharma, one of our customers on the west coast of the United States. This shows that with their CHO cells and their plasmids, they can also start at high culture densities post-electroporation, and due to the high viabilities that they achieved, they also get antibody titers exceeding 500 mg per liter. You note they could maintain their cultures for at least 12 days using a batch-fed process. At the end of that 12-day period, viabilities were well above 60%. I'd like to share just a little bit of data on transient expression of other proteins, or proteins other than conventional monoclonal antibodies. In this particular study, we expressed a diabody using static electroporation to transfect a bicistronic plasmid into CHO-S cells. You can see that in addition to high titers, the MaxCyte process produced protein with very low levels of aggregation. And after column chromatography, you see only 2 very clean bands on the gel corresponding to heavy and light chains. MaxCyte technology is also widely used for vaccine and viral vector production. In this case, a customer is interested in developing a subunit vaccine by expressing the HIV GP145 protein in CHO cells. We performed both small-scale and large-scale transfections with the GP145 expression plasmid into CHO-S cells using static and flow electroporation, respectively. Note that after 10 days in culture, viability in both sets of transfected cells is still near 90%, and consistent with the viability of a parallel culture that we transfected with an antibody control plasmid. As I've shown multiple times throughout this presentation, titers generated via small-scale transfection are highly consistent with titers generated via flow electroporation. I should also mention that these GP145 titers are significantly higher than the titers generated by the customer's conventional transfection protocols, even compared to titers generated in HEK cells. Now, so far, I've been focusing on the benefits of MaxCyte technology for high-titer protein expression in transiently transfected cells. However, I'd also like to point out that MaxCyte offers significant advantages for generating stable pools and clonally derived stable cell lines. High levels of transfection efficiency and high post-transfection viability allow the application of more stringent selection protocols. And due to their high viability, MaxCyte transfected cells typically recover more quickly from selection compared to cells transfected with other methods. In this experiment, we applied selection 1 day after electroporation of CHO-S cells with a bicistronic antibody plasmid. Within 2 weeks, cells recovered from selection and we had a stable pool. At that point, we initiated limited dilution cloning, And after screening about 400 clones, which is a relatively small number, they expanded some of the high-producing clones for further characterization. By the end of 8 weeks, the highest producing clone was then placed into production culture. So again, the entire process from electroporation to a production culture in this experiment took only 8 weeks. If you look at data generated with one of our higher producing clones during culture scale-up, you can see that the titers in that culture exceeded 3 grams per liter. As a point of reference, day 0 in this data set is actually 6 weeks post-electroporation. And then finally, I'd like to mention once again that in addition to our STX instrument, which can transfect up to 2 times 10 to the 10th CHO cells at one time, MaxCyte also offers the VLX instrument, which can process up to 2 times 10 to the 11th cell in a single 30-minute transfection run. Scalability from the STX to the VLX is seamless. Cell type compatibility and ease of operation are also the same as with the STX. And because MaxCyte technology was originally developed for cell therapy applications, the technology, which is supported by a master file with the U.S. Food and Drug Administration, is compatible with GMP bioproduction. So given the capability to generate tens of grams of protein much earlier in the development process, I'd just like to get all of you thinking about the applications that could be streamlined by the MaxCyte process. But at this point in my presentation, I now would like to hand the presentation over to Jerome and Audrey from Valneva, who can tell you about their experiences with the MaxCyte STX.
Thank you. Thank you very much, James. And hello, everybody. I would like first to thank MaxCyte for giving us the opportunity to show our work. Audrey and myself will present to you the work we performed in order to apply the MaxCyte transient expression process to our own CHO cell line. Most of this work will concern fine-tuning on horticulture parameters. First, I will expose to you the context of our need to develop a CHO transient expression process and how we did with the support of MaxCyte. I will also explain to you the limitations that we observed and the parameters that we have to improve. Then Audrey will continue this presentation showing you our experimental results on cellular amplification, optimization, and our work on the impact of cellular aging on transient expression. Finally, we will summarize this work in the conclusion. So, Valneva developed an innovative and powerful method for antibody discovery, so-called VivaScreen. Hence, to support a client and discovery program, Valneva needed to develop its own CHO platform. First of all, we established the following cell bank system based on subclones selected on growth and transfection abilities. Then a stable cell line generation process was established, very convenient for the high amount of protein, but time-consuming. We needed also a rapid process for the production and the screen of map candidates, production of antigen or benchmark antibodies. That's for this purpose that we contacted MaxCyte in order to assess their technology and especially the MaxCyte STX device. With MaxCyte support, few experiments were designed to apply this transfection process on our own CHO cell line. So, as you can see on the schematic representation of the transfection process, you can see on the right, the left, the left that we follow the MaxCyte recommendation concerning cell amplification, applying a cell split the day before the transfection. So we performed a cell split on the day before the transfection. Here, we also assessed different parameters on the transfection day. We were using the pre-established CHO transfection protocol, and then we assessed DNA concentration. We assessed also different kind of vector, tandem vector, that where the light chain and the heavy chain are in the same plasmid, and also the control friction of plasmids that will encode for light chain and heavy chain. And we also assessed the reproducibility of the transfection between large scale and small scale. We also assessed during the fed batch kinetics different kind of fed batch media, and we also assessed different cell density during the fed batch kinetics. Thus, minor adaptations were performed on MaxCyte CHO generic transfection process to adapt it to Valneva's CHO. Here are the titers that we obtain during when we applied this transfection protocol to our own CHO cell line. All the titers that you can see on this graphic were determined by HPLC protein A at day 6, 10, and 13 post-transfection. On the left panel, there is an expression of MAB1 with the co-transfection of the plasmid, encoding for the light and the heavy chain, the expression of the tandem plasmid at small scale and large scale. And on the right panel, the same condition with the MAB2. So, here, what we can see that the titers are varying according to the antibody that is expressed, which is normal. You don't expect to have the same expression for all the IgG1s that you will transfect. Also, the antibody titers obtained are sufficient for our use. Yes, we were expecting to have high titers, but not as much as that. 600 micrograms per ml is really interesting titers. What is very good also is that we were able to achieve the same titers at the different scale. From the large scale and the small scale, the same titers were obtained for both, for the MAB1 and the MAB2. So it's really nice. And what is also very nice for us, that was the co-transfection of plasmids that were not optimized, where we were able to reach really high titers, around 100 micrograms per ml. So that is really nice for us, because it's really important for us in the early stage to be able to screen the MAB candidates. We assess this transfection process on the different banks that we have, the pre-MCB, the master cell bank, and the working cell bank. What is very good is looking at the standard heroes bar. This process appears for us very extremely robust and reliable. And this is the case with MAB. On working cell bank, we were able to reach up to 600 micrograms per ml, which is very good. Now we have a record of 1 gram per liter with this MAB. Despite the good antibody titers that were obtained, some process limitations were identified. The cell density is one of the major problems that we identified. In fact, vanilla CHO density is around 2 million cells per ml on the transfection day, as you can see on the graphic just behind. Yeah, here you can see the variable cell density along the cell amplification. We are performing a cell split day 1 before the transfection, and on transfection day, the variable cell density will be around 2 million cells per ml. That will impact that for a transfection of 1 billion cells, we will need around 5 liters of cell amplification. This will limit the number of large scales that we can handle altogether, and naturally, it will impact the production cost and handling easiness. Another thing that we observed, that we know, it is well known that cell aging could affect different parameters. And what was our question was that does cell aging will affect the antibody titers obtained by transient expression? To assess this, we decided to perform some transfection along cell aging. In this way, the transfection was performed at different passages, and we assessed the titers that were obtained. That will allow us to limit the variation between the transfection and establish the best window for the transfection efficiency. Naturally, the overall objective of this work was to reduce the time and volume of cellular amplification, test the impact of cellular aging on cell growth and antibody production. We want to improve antibody production and process robustness while reducing working time. And now, Audrey Angel will expose to you our experimental results.
Thank you, Jérôme. So in the first time, I will talk about the cellular amplification optimization. So as a cell concentration of very large quantity of cell is necessary to perform the transfection with the MaxCyte STX device, the cell density was identified as a limiting parameter during our cell amplification process, leading to a large volume to be handled. Furthermore, to get the best transfection efficiency, it is known that the cells have to be transfected in the exponential phase. So our objectives are here to search the best cellular amplification condition to get the culture in exponential phase and with high density. And then we will validate this condition for transfection, the best transfection condition. So we first test a supplement that boosts the cell growth. So we let the cell grow over 1 week with or without this supplement, starting from different seeding densities ranging from 0.25 to 1 million cells per mil. And what we see concerning the addition of the supplement is a significant cell density increase with the supplemented medium. And furthermore, the exponential phase is longer in the supplemented medium. And we see no significant viability difference between both conditions with or without the supplement. So this supplement was very interesting for us, and we considered it to be further explored. Then, in terms of the initial seeding density, we see that at day 3 in the supplemented medium, the cells are still in their exponential phase, and very high viable cell density are reached when initially seeded at 0.5 or 0.75 million cells per mil. So this process for amplification is very suitable to start on Friday for transfection on Monday. Then we will validate this condition with transfection. So before the transfection, we validate again the benefit of the supplement addition in the cell amplification. So we see here again a 2.5-fold increase in the cell density after 3 days of addition of this supplement. After transfection of a MAB encoding vector, we see no impact on MAB titers, no impact of the amplification process. So the new amplification process clearly enhances the cell density on the transfection day, so it facilitates the cell culture handling by reducing the volume, and of course it reduces the cost, and it does not impact the antibody production. The second part of the results are about the effect of the cellular aging on transient expression. So the Valnivas show cell line exhibits a very regular and constant growth over 60 days. That means around 25 passages. So we performed a series of 7 transfections every 3 passages with both GFP and MAB encoding vectors. About transfection, GFP transfection efficiency was determined by flow cytometry. We see that we have a very efficient transfection with more than 95% of fluorescent cells detected. And this transfection efficiency is very stable along passages. What MAB titers are concerned, we see that the cell aging effect is more obvious. Actually, we observe at the 10 that high antibody titers are obtained around 800 micrograms per mil. That is very good. But this MAB production drops continuously beyond P13. So we can determine an optimal production window between P7 and P13 included. As a conclusion, to sum up our work, we were able to easily adapt the max transfection process to our Valnivas show cell line, reaching high antibody titers, varying according to the antibody express, of course, with almost the same antibody titers at small and large scales. In a second time, we implemented some optimization on routine culture to enhance the production capabilities and robustness. We successfully reduced the amplification time and final volume of the cell suspension. The new amplification process allows thereby to enhance the cell density on the transfection day, thus facilitating the cell culture handling by reducing the volume, namely 2.5 times less volume, and this does not impact the antibody production. And concerning the cell aging effect, GFP transfection efficiency is stable along passages, but MAB expression is not. So an optimal transfection window for MAB production was established between P7 and P13. That's it. So thank you very much.
Great. Thank you very much, Jerome and Audrey. Before we open up the webinar to questions, I just want to present a few of the key conclusions from this webinar, points I want you to take home. First of all, with MaxCyte, the high viability and high transfection efficiency that we can achieve, particularly with CHO cells, leads to high protein expression. The variety of protocols that are available for different cell types means that development can be conducted in the manufacturing cell line of choice. You don't have to use hex cells. You can do all your upfront work in CHO cells. Improved expression and reduced culture volumes by transfecting cells at high density and culturing cells at high density after transfection means reduced manufacturing costs. And the bottom line is that the MaxCyte transfection process leads to a simple, reproducible, cost-effective transfection method for development and manufacturing. So, again, I'd like to thank all of you who listened to us today, and in the remaining few minutes of our webinar, we'd like to get to some of the questions that you posted.
Thank you very much, Jim, Jerome, and Audrey. Yes, we have quite a few questions that are coming in. This person asks whether this has been used for cGMP production for early phase materials and whether it's been accepted for phase 1 trial use in the European Union and U.S.
So what I can tell you is this technology was originally developed for cell therapy applications, and it still is used for cell therapy. We take cells from patients, we transfect them with molecules to make them more immunogenic, to alter their biological properties. We give them back to patients for treating cancer and other diseases. So as I mentioned earlier in our presentation, we do have a master file with the FDA. It's GMP compliant. And in terms of where we are in terms of producing GMP-compatible materials, what I would probably do is refer you to regulatory experts in our company who can answer that question more completely. If you look at the screen right now where it says info@maxcyte.com, please give me your contact information and we can handle those regulatory questions specifically, try to address your interest specifically.
And another related question, do you have examples where MaxCyte has been used for phase 1 or in the market?
That basically relates to the last question. So again, we have regulatory experts who are not participating in the webinar today, and they can give you more details on the regulatory applicability of our technology. So please send your specific questions into maxcyte.com for those answers.
Okay, and a question for Jerome and Audrey. What were CHO cells supplemented with to achieve the high log phase density?
Using a commercially available cell boost.
All right. Another question. Are stable clones derived from MaxCyte transfection process more or less stable than more conventionally selected clones? For example, copy number effect.
So, we don't have a lot of data on actual copy numbers. What we've seen in terms of the stability of the clones, if you use a low level of selection after generating the stable clones, stability is at least as good as you see with other transfection methods. But what I was trying to point out in my portion of the webinar is that you can actually apply higher levels of selection earlier on because you are getting higher transfection efficiency and higher viability to start with. So I think that's one of the key advantages there for making stable clones. You can get more DNA into the cell early on, you can apply higher selection, and you don't have to screen as many clones to find a high-selecting clone.
All right, a question specific for Jerome. Which cell boost, 1 to 6?
Well, I'm not allowed to say which cell boost we are using. The point is that you will have to assess a different cell boost to really adapt it to your CHO. There is different kind of CHO, and they will definitely have different kind of behavior in the case we are using one or another one. So I will suggest to use different kind of cell boost and to look for the right one to your CHO.
One thing I would add to that is that the data we presented, at least from MaxCyte, was done with a fairly minimal level of process development. There are a lot of sophisticated approaches that could be employed to further boost the titers that we showed. If you do a spent media analysis, you supplement with specific amino acids, you could probably maintain the cultures even longer than what we showed. So what we did was really just a minimum level of process development. Sounds like Jerome and Audrey may have taken it a little bit further, but those who are sophisticated in protein production methodologies could probably improve further on the protein level that we showed in this webinar. And I saw a question about plasmids. Maybe I could talk about that a little bit, and Jerome could also add to it. One question was the size of the plasmids that we transfect. Our bicistronic plasmid about 12 kb, but we really don't, we've never really seen an upper limit to the size of the plasmid that can be transfected by electroporation. As I pointed out earlier, the technology is also compatible with large proteins, other macromolecules, so you're creating significant temporary openings in the cell membrane that can be used to transfect very large plasmids. And you can also co-transfect heavy and light chains. I know Jerome and Audrey show that they got better results by using a tandem expression plasmid. But we do have customers co-transfecting heavy and light chains and still getting titers over a gram per liter. Sometimes altering the ratio of the heavy and light chains can also improve your results. And Jerome, did you have anything that you wanted to add to the relative virtues of co-transfection versus tandem vectors?
Well, for the co-transfection, we haven't assessed all the conditions. We were using just a ratio of 2 light chain for 1 heavy chain. And, well, that is true that, well, there's still some work to do. We haven't assessed also different kind of plasmids, meaning that the one that is the highest size that we were using is 12 kb. We have used from 6 to 12 without see any effect on viability post-transfection. So, well, I don't know over 12 kb, but for up to 12 kb, it's really nice.
The main optimization you really need to do is find the actual total amount of DNA that's right for your particular cell and plasmid. You put in too little DNA, the expression level might be low. Too much DNA is going to be toxic to the cell. So really the only optimization you need to do is test a few different concentrations of total DNA. You do that using the OC400, and then you could use that same concentration to scale up the process in the CL2 for large-scale transfection. And then, of course, in the case of a co-transfection, as we talked about, you might also want to optimize the relative ratio of the two plasmids.
So there is a very good question about why we are not seeing any plasmid dilution effect after several days. Well, he's already answered in this question. It's probably from the high initial copy number that is integrated inside the cells. When we are looking at the cell impact, cell aging impact, I mean, that the transfection ability was still the same, but around 99–95% for along the cell amplification and cell aging. But the titers that we obtained were decreasing after 13 passages. That means probably that the copy number that was integrated when the cell was getting older was less than it was before, because the GFP, a few copy will trigger the expression, and so you will see the transfection ability around 95–99%. But this meaning so that probably the copy number is less when less integrated when the cell is going to be old. That's what we were seeking for our own cell line.
Okay, we also have a question regarding the samples. This person says, I have no experience with vaccine research and development. So he wants to know, do cells need to be in suspension, or can they be both suspended and adherent?
So the technology works with either suspension-adapted cell or adherent cells. In both cases, the cells are harvested and put into our buffer during electroporation. But afterwards, the adherent cells can be grown in T-flasks, they can be grown in cell factories, they can be grown on microcarriers, and of course the suspension cells can be grown in wave bags, shake flasks, whatever you normally use for suspension culture. So it's compatible with any type of cell.
All right, a question from Krista. What do you have to do to scale from the STX instrument to the VLX? Is the transfection process different?
The same process, you just prepare more cells and add a proportionally larger amount of DNA, but whatever conditions you optimize in terms of the DNA concentration and the plasmid ratios using the STX, those exact same conditions can be applied to the VLX just at larger scale, and you should get very consistent results.
All right, and Saida asks, any preference for a commercial plasmid to be used for antibody production?
Our customers use a wide variety of plasmids. Most of them have CMV promoters. There are a number of companies that sell plasmids for protein expression in mammalian cells. So we don't have any special requirements. Jerome, do you have any recommendations for plasmid backbones?
No, we assess the different kind of backbone also, and using a CMV promoter also. So yeah, I would say, according to your cell line, you will have to look for the right backbone with the suitable promoter. That's it.
And also, I mentioned earlier, we can do this with insect cells too, and in that case, of course, you'd have to use a insect cell specific promoter.
James, we have a question for you from Gregor. Can you explain the high viability after electroporation, which is normally a trade-off?
Well, I think some people in the past have used other electroporation devices where you have to try to guess at the electroporation parameters, try to find the right capacitance, voltage, etc. And if you apply the wrong conditions, it is very easy to lower the viability significantly. So we put a lot of effort into optimizing our electroporation protocols. We have people at MaxCyte who've been with the company for over 12 years, focusing all of their efforts on developing those protocols. Also, I want to point out this technology was developed for clinical applications. So we've come at electroporation from kind of a different angle. When you're taking cells from patients, it's very critical to maintain high viability. Those cells are very precious, and you want to give them back to the patient with minimal perturbation of the normal biological activity. So in addition to the optimized protocols, we've also put a lot of effort into optimizing our buffer formulation. So I think all of those factors really make this quite a bit different from other electroporation devices that you may have worked with, and that's what really is the key to our high viability as well as high efficiency.
And Driss asks, how cost-effective is this system?
We have people who can give you very detailed cost rundowns on how this will compare to other technologies. Again, since I'm the scientific expert here, I'm not the best one to address the cost effectiveness, but what I would encourage you to do, again, is to go to the info@maxcyte.com, tell us what kind of information you're looking at, what your current process is, and we can help you walk through the cost effectiveness.
All right, another question. Can you transfect the heavy and light chains at the same time? And if you can, how much DNA is required?
So again, the total amount of DNA is going to be the same, whether you're using a single plasmid or co-transfecting. So if you're using 1 to 2 micrograms per million cells, it's going to be the same, whether it's 1 plasmid or 2 plasmid. As we were discussing earlier, if you want to play around with the relative ratio of those 2 plasmids, a lot of our customers add a little bit of excess light chain to improve secretion. That can also help your titers. But the total amount of DNA is going to be the same. It's 1 plasmid versus 2 plasmids.
And Claudia asks, do you have any experience in his-tag recombinant protein production? She's having problems with very low yield, and with an FC tag, she doesn't have yield at all.
The answer is yes. We have experience with both of those 2 types of tags. Again, tell me what you're doing now. Show me some of your current results, and I can tell you whether or not we think we can give you a significant improvement. So just reach out to info@maxcyte.com, and we'll get back to you very shortly.
And we're getting up to the hour, but time for one more question. What media and feeding schedule did you use to produce optimum antibody titers?
So I can tell you, in our case at MaxCyte, it's a mixture for the feed formulation. It's just a mixture of amino acids and sugars and other common supplements that we buy from the major cell culture suppliers. So it's I don't know I can't give you the exact formulation off the top of my head, but it's a very conventional feed strategy. And Jerome, I don't know if you have anything special you want to mention about your feed strategy.
Well, we assessed different kind of fed batch. The one that was giving us the best results was the one that was recommended by MaxCyte. So we are using currently your fed batch.
I'll also point out that we also work very closely with our customers after they acquire the technology to help them implement our feeding strategies and media recommendations. So we provide a lot of support after purchase in terms of helping to translate all these technologies into your facility. But there's nothing really special about the media or feed that we're using. It's all commercially available.
Excellent. Well, I see we've come up on 12 o'clock, so we probably should wrap up. I'd like to thank you, James, Jerome, Audrey, for your presentations today, Maxite for allowing us to do this webinar and sponsoring it for us. With that, I'd like to thank all of you who have spent some time with us today. I hope the information has been helpful and interesting for you, and I look forward to seeing you at future Cambridge Health Tech Institute Global Web Symposia. Thank you all, and have a great day.